Okay, thanks a lot for the introduction and thanks a lot for the invitation, and it's really a privilege to be here for me, particularly coming from Tenerife, and you are most welcome to, in the next few years to come and do this in Tenerife. For us, for someone like me in the Canaries, it's even more important to be in a consortium like this, because we are, somewhere there isolated, so it's a benefit from me, and I really hope that I can actually give back something, at least in terms of collaborations, and this is why we're here. So these are all my affiliations, and today what I want to do is to try to condense a little bit of the research we've been doing in the last few years, focusing on the development of new ALS mouse models. Whoa. Dr. Osta introduced a little bit of ALS. What I want to say is that 90% of it is sporadic, but 10% still, or around 10% is still familial. And we use, as almost everyone in this room, these genetics as an entry route to try to drill into novel disease mechanisms. The fact is that SOD1 was the first gene isolated as causative of ALS. This was 30, 40 years ago, and we still don't really know how SOD1 mutations really cause ALS. And this is arguably the case for all the other genes that come afterwards. Perhaps not C9, but we won't talk about that today. So in terms of the genetics, there are, since SOD1, there's been a flurry of genes causative of ALS, and today we're going to focus on particularly a couple of them, or three, TDP43, FAS, and SOD1, okay? So Dr. Osta focused her talk on the really workhorse of the SOD1 community that has been the SOD1 G93A model. It's an absolutely amazing model. It really has almost all the features of ALS in terms of having end-stage upper and lower motor neuron degeneration. However, there is some disquiet in the community as a lot of the trials based on the SOD1 mouse have really failed into clinical trials. And there are many different possibilities for that. One is that the model itself is great as a SOD1 model, but perhaps not as a model for other ALS genes, could be. But also that it massively overexpressed the SOD1 protein, and overexpression of SOD1 wild-type protein also have some features, arguably not ALS, but it gives some pathology. So since SOD1, there have been a lot of other genes, and a lot of these other genes that have been causative of ALS, like TDP43 or FAS, are RNA-binding proteins that are really exquisitely dosage sensitive, meaning that the transgenic models are really not the way forward. And why? Is because it's very difficult to disentangle the toxicity from overexpression from the toxicity produced by a pathogenic mutation. So a few years back, and this is a collaborative effort not only from me, but from the labs of Lizzie Fisher, Linda Greensmith, and Pietro Fratta at Queen Square at ION in London at UCL. And what we decided was to start creating new novel models of ALS genes, but creating knock-in models, creating mutations that don't overexpress. So we first started a long time ago where CRISPR technology wasn't there, not even in the horizon. So we started with this old stuff that was ENU mutagenesis. So this is an old trick for mutagenesis, and it can work in the mouse in the sense that we had ENU archives around the world, one in Harwell in UK where it was, but also in Japan and in Germany. And what you can do with those was to screen those archives for mutations in your genes of interest. And these were mice that were injected with ENU, then crossed with wild-type females, and then you get those F1s and you kill them. and you keep the sperm, and then you keep the DNA in your screen, and it's a way of creating point mutations by random mutagenesis. So the mutations choose you, not you choosing the mutations as we do now with CRISPR. So we've done that with a few of these genes, and then clearly after the advancement of CRISPR, we are moving on to CRISPR. However, it really has its advantages to work with ENU mutagenesis because sometimes the things that you're not looking for are useful. But anyway, here we go. So the first one we start with, it was SOD1. So we started with ENU mutagenesis, and we were lucky there that the archive in the UK had a mutation that was identical to a human mutation. So this was the D83G. That is a mutation that is causative of ALS in patients. It's an interesting mutation because it affects the 83, the zinc, it's one of the aspartic 83 is one of the two zinc binding residues. So it really kills the function of the protein, okay? So there are two main mutations in SOD1 that cause ALS. Some actually interfere and reduce the dismutase activity, and some don't. And this one is clearly one of the ones that do. So this was the first knock-in, or if you want, the first mouse model carrying a point mutation in the SOD1 gene, and we went on and characterized it, and to characterize, and as a lot of you know, characterizing a new mouse strain is really a lot of work. So this is the work through a lot of people and a lot of years. So we only found a phenotype in the homozygotes. This is not the case as in patients. The patients, the SOD1 mutations and the great majority of ALS mutations are dominant. This is not the case here. But we did find some motor neuron degeneration, and okay, I can see it here. Is there oh, it's here. Okay, go slow. But so what we can see is motor neuron degeneration. There is a wave of motor neuron degeneration from 6 weeks of age to 15 weeks of age, but then really, really surprising to us, it doesn't really progress. And why it's really surprising is because those mice, phenotypically, motor-wise, they decline dramatically. We also look at gliosis, and we also look at the upper motor neurons a little bit later, and the fact was that the motor neuron degeneration didn't really explain the worsening of the phenotype. So what we found, looking at the neuromuscular junctions, was that what they were having was a really strong peripheral neuropathy. Ooh. And actually, looking in the literature, we always see that the SOD1 knockout. So there is that the loss of function in SOD1 has nothing really to do with the disease, but actually the SOD1 knockouts, if you look at the literature, they really have a really, really strong peripheral neuropathy. So the neuromuscular junctions are retracting, but the motor neurons are still there. And this is what happened in these mice. So these mice have a dual, if you want, hit. One hit, the motor neuron degeneration, that is caused purely by a gain of function in SOD1. And why is that? It's because the SOD1 knockouts don't have any motor neuron degeneration. But what really drives the phenotype after that is the SOD1 loss of function. Okay? So these are a good model to try to understand the consequence of central versus periphery in ALS. Okay? And these mice are available through JAX and at EMA, the European repository. So moving on, over the last few years, we've been really focusing on working on TDP43. I don't have a slide, but I'm going to explain it a little bit. TDP43 is the amyloid or the synuclein of ALS. So it's the main component of the inclusions in patients. As well, mutations, missense mutations in the gene encoding TDP43 that is called TARDBP are causative both of ALS and or frontotemporal dementia. So we started looking at, again, at this CIIEN archive, and we found a lot of mutations in archive in the UK and in Japan. But also now the CRISPR was becoming available, so we made a CRISPR knockout, a CRISPR knock-in. So we introduced this mutation. So I'm gonna focusing on these two, and why is because we went on and studied in detail the molecular mechanisms underlying these mutations. So what was really surprising, actually, from the beginning was that we could see a complete opposite effect in terms of the splicing function of TDP43. I should say that TDP43 is an RNA binding protein, and that has many functions in RNA metabolism, but one of them is to control the splicing of a lot of cassette exons. So what we found at the beginning was that it was clear that one mutation that was in the RNA binding domain, the second RNA binding domain, gave a loss of function mutation, and that was due to really diminish RNA binding capacity. Whereas a mutation in the C terminal actually gave the opposite. It gave a gain of splicing function. And how we did that, we used a reporter with CFTR that actually is used throughout the literature, and it's well known as a target for TDP43 splicing. But we went on and do in-depth RNA-seq and found that the signature, the transcription signature of both mutations were opposite. And this was surprising. And why it was surprising was because looking at the transgenic models that are out there for TDP43, that's not the case, because they do have an element of loss of function. Whereas here, what we found was that with two different C-terminal mutations, what we did have was a pure gain of function, at least in terms of the splicing. And how we explain that is possibly because TDP43 is an RNA binding protein that autoregulates itself. And by overexpressing TDP43, human TDP43, the endogenous mouse TDP43 gets down, and that might give some loss of function, okay? So looking in depth at the RNA-seq analysis, we realized that there were certain transcripts that gave a unique signature of this gain of function. And we named them skiptic exons. And why we named them skiptic exons was because a few years before, there were, as a signature of loss of function in TDP43, they were the opposite exons named skiptic exons. Those were exons that were skipped when, well, the skiptics are, well, I'll explain a little bit, because if not, so we call it skiptic because they are excluded, okay? So these are genes that are usually, at least in the spinal cord, they are constitutively expressed, and when you have a TDP43 gain of function, they get excluded, okay? And this leads to nonsense-mediated decay, and at least at the RNA-seq data, we see a decrease in the expression of those transcripts. Let's face it, I mean, there are 47 of those that we could find, and this is in the whole exons of the whole genome, so they are a tiny bit, okay? But what we see here is at least a molecular signature of TDP43 gain of function that is unique and is not really the opposite of a loss of function. These are genes that are, or exons that are coming in and out that only happen when you have TDP43 gain of function, okay? So these are really interesting genes, and some of them, 4 or 5 of them, are E3 ubiquitin ligases, and they really are genes, so the transcripts that include those genes that have pleiotropic effects, and we are working a little bit on that. And this is the work together with Pietro Fratta's laboratory in Institute of Neurology. So looking at the mice themselves, we realized early on that this, I should say that this is the MC23K mutation that is in here. So the mutation is in here, but I should stress here that this is a mutation that is not found in human patients. So this is a mutation that, through ENU mutagenesis, the mutation found us, and it's not in human patients. However, the mice that are homozygote for this mutation actually have motor neuron degeneration, and they have, now it's lost. Okay, I use this. So they have around 30% of motor neuron degeneration that is progressive, and this is around 2 years of age. So these mice are normal until, at least in terms of motor function, until around 1 year of age, and then they start to decline, and they reach around 20–25% motor neuron loss by the age of 2 years. So this is a progressive model that don't really develop full-blown ALS, but for us it's useful to start to drill into disease mechanisms early on. Again, interestingly, they don't have TDP43 inclusions. They are inclusions in the spinal cord, but they are TDP43 negative despite having TDP43 mutations, although they are P62 and ubiquitin positive. So what we're trying to do at the moment is to use cellular models from these 3 strains of mice that we have that carry this allelic series of mutations that allow us to distinguish between loss and gain of function and compared to a pathogenic mutation. And what we are looking is at the biochemistry of these TDP43. And as example of this kind of work that we are doing at the moment, here you can see that TDP43 gets precipitated in the mutant, in this case it's MES, by this chemical called the isox that I explained a little bit, a lot more than the wild type. And this is the case both with the QT31K mutation, but also with these other MC23K mutations. So the isox is this chemical that was identified by the McKnight laboratory. And what it does is that it forms crystals, and it precipitates particularly proteins like prion-like domains with low complexity that are prone to do liquid-liquid phase separation. So it's, if you want, a proxy for liquid-liquid phase separation. So what we see here is that C-terminal mutations, not only 1, but 2 of them actually impact on the ability of TDP43 to get precipitated by the B-isox. Also, what we are seeing is that we're using DSG as a cross-linking, and what we're looking at the oligomers, at the multimers, if you want, of TDP43, and what we see is that there is a unique band here on the mutants that we are currently trying to identify. And ideally, what we would want to do is try to link those with this gain of splicing function, and that's the type of work we are working at the moment. We're doing at the moment. So, as a brief summary of the work on TDP43, so we have a allelic series of mutations that, of course, are freely available. All the mice we make are freely available to the community. However, these TDP43 came, or particularly 2 of them, came from a Japanese mutagenesis archive, and the MTAs in Japan are a little bit hard, so we are trying to get them to agree to pull them on the free domain. It's important that if you want to use these mice, you are most welcome. I have them in Tenerife, so they are yours. But a little bit of an issue, it's more than a little bit, is that at least one of the mutations needs to be on a hybrid background to be able to survive embryonic development. And this is the mutants that develop motor neuron degeneration. So we need to maintain 2 colonies, which is a bit expensive. Okay? So for completeness, I really wanted to talk about FAS. This is not my work. I'm involved on it, but it's really Lise Fischer's and Annie Devoy's work. But I wanted to touch on these because these are another great model of knock-in. This particular model have a frame shift mutation that is causative of ALS and cause juvenile ALS in patients, and it creates a frame shift that allows to create an antibody that actually follows only the mutant protein, and that is a monoclonal antibody that comes with the mice. So it's a good mice. The phenotype is quite similar to the MC23K LCD TDP43 mutant, and they have progressive motor neuron degeneration that only happens after 1.5, 5 years or so. So I'm finishing. Just an example of comparison between transgenics and knock-ins, but I wanted to finish on what we are working, another point that we're working on the moment. We are trying to fully humanize different genes, and fully humanize, at least what we are trying to do is to get rid of the mouse gene, exons and introns, and introduce the human gene. So with Lizzie Fisher, and this again is a work with Lizzie mainly, they have succeeded that with FAS, and now we are in the process of doing it with TDP43, and as from last week, we had some ES cells that seem they are expressing human TDP43. These are a human-specific antibody, and here we can see that there is a TDP43 that is only expressed in the ES cells but not in the MEFs. So we have some correctly targeted ES cells, we think, and now we need to pray to get them germline, which will be a big deal. But at least that's what we're trying to do. And by the way, this is the approach we took is to humanize from the ATG to the stop codon, including all the introns in between, leaving the mouse promoter and the 3' UTR as mouse, because there are some reports that say that that might be an issue in terms of embryonic development. Just the two main points, and you can read them if you want. For me, the main point here is that you can really use the knock-in models to try to drill into disease mechanism, but mainly the main point that is not there is that we need to embrace the complexity of ALS, and that we need to try to work with different models and not just focusing on the SOD1 gene 83A. And I think that is something that the community is moving with us at the moment. Just to finish with the acknowledgements, this is a huge collaborative effort. So our lab in Tenerife, but also at MRC Harwell, where I used to be for a long time, and ION UCL. Thanks a lot, and take any questions. Thank you.
Thank you, Abraham, for your comprehensive review of the animal mouse models. Time for questions. True, yes, Ramon?
And if it is, what would be your hypothesis for the selectivity?
It's not. So it's everywhere. At least this gain of function, we see it in fibroblasts. I didn't say, which is a huge point. It's also present in fibroblasts, TDP43 mutant fibroblasts from patients, at least some of these skiptic exons. It's everywhere. And I guess it's a big issue with all ALS genes. They are all ubiquitously expressed as the great majority of neurodegenerative genes, and why they cause the disease where they are, I have no idea. There are some isoforms that might be specific, at least from the TDP43. There are some isoforms, as you know, or some cleavage forms that are present in ALS. That are unique from the spinal cord, but so far cannot really say.
Thank you very much.
Sure.
Hi, congratulations, great talk. I was wondering, the mutations you mentioned in the low complexity domain of TDP43, you mentioned that they do not coincide with any of the human mutations, but I was wondering, or do they?
One of them doesn't, which is the MC23K, what I call the LCD mutant, but the other one that we made with CRISPR, the QT31K, it's a pathogenic mutation in patients.
And that's the one that has the...
Both of them do the same. both of them do the same. So there are no difference in terms of the biochemistry events that we are seeing. Both of them really do the same in terms of splicing gain of function and what I presented here in terms of the propensity to get down with B isocs and also with the oligomer, which give us more credibility to the results, if you want.
So what do you think? What you think is happening is that it's impeding or facilitating the movement to the loci or to the active sites for the splicing because of the phase separation, or? So
we have no idea. We did a lot of analysis. We did CLIP for this EMBO paper, and what we found was that it seemed to bind the same regions. CLIP is not necessarily a quantitative platform, if you want, but at least the binding sites are very similar. However, the gain of function mutations, the C-terminal mutations, seem to bind 5' quite a lot to the exons that give a gain of function. So we think there might be some element there, but we really need to drill in a lot more to really understand it.
Okay. And finally, it was very nice to have the overview of the mouse models from the highly penetrant SOD, then the TDP43 infused that you mentioned. But I was curious, what about the C9? Are there models, and are they good as animal models of ALS or not?
They are a lot of models. Some are good, some are not. I didn't talk to them about them because I'm not actively working on them, but I'm involved with the humanization of some of them as well. There is a particular model that is starting to be very useful to the community. However, it has a problem that some mice have a very strong phenotype and some mice don't. And no one really knows why. These are from isogenic backgrounds, and some mice come on to develop full-blown neurodegeneration, very acute, and some don't. And that really, as a preclinical model, gives you a really big headache because you don't really know which ones are going to develop these acute phenotypes. So I think at this stage, the jury is out as to any other mouse model to compete with the SOD1 gene 83A as the workhorse for preclinical studies in ALS. We are getting there. Some TDP43 models, some fast models, also some C9 models. Again, these are transgenics, viral models as well. We are getting there, but it's slow. Okay? Thank you.